A big hello to all my wonderful NEET aspirants out there. Children, I hope you're doing really great. Your preparation is going really good. Today, I am here with NEET mock test paper 3 solution, that is for botany subject. So, I hope those of you who find difficulty, who found difficulty while solving the questions from the mock test, do not worry. Here I am with the solution. I hope I could be able to make you understand certain concepts as well. Yes? So, let's go ahead with the first question that we have. That is, the most important function of inflorescence is to help in what? Inflorescence, that is flowering, that is group of flowers. What is the use of this? What is the most important function of inflorescence? This is a very easy question. Increasing the length of the plant, attracting animals for cross-pollination, dispersal of seeds, or release of nectar. So obviously it is for when you wear something big or things like that. It's basically for what? Making others know that hey look, here I am, and things like that. Similarly, in the case of flowers, they cannot call out anyone. So basically they have to be big, they have to be pretty, okay, to attract animals for what? Cross-pollination, my dear children. So the most important function of inflorescence is to attract what? Animals for cross-pollination to happen. Cool? Next, DNA replication occurs during prophase, metaphase, anaphase, or interphase. So, you know, DNA replication happens during which phase? So, in the whole cell cycle, there is interphase, okay, and there is M phase. This is the M phase and that is the interphase. Interphase is again divided into 3 phases. One is G1 phase, one is S phase, and the other one is G2 phase. Now, S phase is the one which is also known as synthesis phase. What do you call it? Synthesis phase. Why, ma'am? Because this is where DNA replication takes place. This is where DNA synthesis takes place, my dear children. You want to complicate. Basically, if there is one cell and this cell has to divide into 2, obviously whatever content is there in the cell, it also has to divide into 2. That means I have to make a duplicate copy of itself, right? So all this duplication, all this replication happens in the S phase of the interphase. So DNA replication occurs during prophase, metaphase, anaphase, or the interphase. Your answer is D interphase. Okay. Where in interphase? S phase. Now, housefly belongs to the family what? Hominidae, Mucidae, Anacardiaceae, and Felidae. What is the answer for this? Housefly, you call it musca, right? So basically it is Mucidae. It is what? Mucidae. B. Housefly belongs to the family Mucidae. And I also want to add up one more thing over here. That is interphase, my dear children. It goes up to 23 hours, and 1 hour is where the cell, spends its time for actual division. Okay, so 23 hours there, 1 hour over here. Total cycle, it is 24 hours in the case of human cell. Okay. Now, going ahead. How many of the following statements are correct? Okay, let's see how many of them are correct. Conjoined vascular bundles are not present in monocot stem, as in xylem and phloem together. Okay, that is what you mean by conjoined. So, conjoined vascular bundles are not present in monocot stem. Okay, next thing. Casparian strips of endodermis are made up of cutin. I hope it is made up, you know that it is made up of suberin, not cutin. Now, pith is large and well developed in monocot roots, my dear children. Is the pith large and well developed in monocot roots? What do you think? Yes, it is. The pith is actually large and it is very well developed in monocot roots. Trichome helps in promoting water loss due to transpiration. Trichome, which are root hair-like structures which are there, okay? They are hair-like structures which arises, my dear children. So basically, what is the use of trichome which is there? Okay, what is what is the use of this hair-like structure which is there on the plant, my dear children? Basically, it is going to help in reflection of light, sunlight away, and also it does not allow transpiration to happen, or basically it would stop transpiration to happen. It will not encourage the plant to lose a lot of water. So trichome helps in promoting water loss due to transpiration would be wrong, because it does not promote, it depromotes. It says that do not lose water. So this is wrong. This is wrong, because over here it should be suberin, and over here conjoined vascular bundles are not present in monocot stem. That is also wrong, because it is actually present xylem and phloem together in the case of monocot stem. So this is also wrong. What is correct? Only this. Pith is large and well developed in monocot root. So your only, how many of the following statements are correct? Only one of them is correct. Okay. Now, going ahead, which part would be more suitable for raising virus-free plants for micropropagation? What do you mean by micropropagation? For example, I have a plant, okay, I want to make multiple copies of it, but I cannot rely on sexual reproduction method. So what do? I take a tip of this particular plant, certain cells, a group of cells, that is a tissue, and now this particular part is known as X plant because it was once a part of the plant, that's why it's now X plant. Okay, I'm going to put it in some medium where it will be dividing and increasing in structure, number, yeah, and then I'm going to put it in some hormonal solution such that it will be giving roots and the shoots and stuff like that. This is known as tissue culture, and with the help of this I can micro-propagate, I can propagate a large number of plants, my dear children. So if I want that the plants should be virus free, where should I be taking the parts from? The X plant should be from which part of the plant? Two parts. One is apical meristem, okay, and the another one is axillary meristem. Axillary meristem, both of them are meristem. Meristem is basically a bunch of meristem is a tissue. Basically it is made up of meristematic cells, the one that has the ability to divide, my dear children. So you have apical meristem at the apex, yeah, and axillary meristem towards the nodal regions through where the branches and everything comes off. Okay, so I can take the axillary meristem or the apical meristem for micropropagation such that I would be getting virus free plants. Always remember. So bark, vascular tissue, meristem, node, your answer is meristem. Cool? Now going ahead to the next thing. Dash do not grow up in polluted areas. So dash is a pollution indicator as well because it does not grow in the polluted areas, and it was there. Industrial melanism, if you know the experiment, okay, done during Charles Darwin's time, my dear children, you would be able to answer this particular question as well. So what is that thing? It was lichen which was not able to grow in polluted areas. Remember, so lichen which is there, which is a combination of algae and fungi, cannot be growing in areas where it is too much polluted, my dear children. So it's not bacteria, it's not fungi, it's not bryophytes. Your answer for this question is what? Lichen. Lichen do not grow in polluted areas. Cool? Works? Chalo. So they are what? Pollution indicators. Seventh, which of the following organisms are decomposers? Terrace, bacteria, saprophytic fungi, both B and C. So I always make children remember decomposers. Decomposers are the one that feeds on dead and decaying matter, my dear children, and it includes FB, Facebook. F stands for fungi and B stands for bacteria. F and B. Okay, so which of the following organisms are decomposers? Terrace, bacteria, saprophytic fungi, or both B and C? Your answer would be both B and C. What are saprophytic? Saprophytic is the one that feeds on dead and decaying things only. So that is true. Yes? Cool? Chalo. Yeah. Now, in Griffith's experiment, what was happening? R strain transformed to S strain and became virulent. R strain transformed to S strain and lost virulence. S strain transformed to R strain and became virulent. S strain transformed to R strain and lost virulence. What is your answer for this question? So, initially, please remember, okay, he took a strain of a bacteria that is pneumococcus, okay, sorry, Streptococcus pneumoniae, not pneumococcus, Streptococcus pneumoniae, yeah, and this particular bacteria had two strains. One was the R strain, another one was the S strain. R stands for rough and S stands for smooth. Cool? Now what exactly happened? What made it rough? What made it smooth? There was an extra layer surrounding it, which is why it was smooth. And you know what? Because of this, it was virulent. That means any organism that was infected with the S strain of the bacteria would be dying off. R strain did not cause any problems. It was non-virulent. So I'll say it is non-virulent. Now my dear children, you have three different scenarios, okay, or more than that. I put R strain and into a mice. I find that the mice lives. The mice does not die because it is not going to cause any trouble. I'm going to put the S strain plus the mice. What will happen? The mice will die because S1 is a virulent form. Now what did I do is that I heat killed. That means I treated the S strain with heat. So I'll write heat killed S strain. Heat killed S strain. And then I put it in the mice. Since because of heat some changes were happening to the smooth strain, which is why it was not virulent anymore, which is why the mice lived. Now the fourth scenario was I took the R strain, the one that does not cause any problem, plus the heat killed S strain, again the one that did not cause any problem here, and I put it in the mice. But to my surprise, I found that the mice died. Why, my dear children? Probably because something was getting transferred from here to here. That is, from heat killed S strain, something was transferred to R strain, and now this R strain behaved like an S strain. Behaved like an S strain and killed the mice, my dear children. Okay, so in Griffith experiment, R strain transformed to S strain and became virulent. Yes, because there was something being passed down from here to here. What was it? It was actually the DNA. About this, later they came to know when Hershey and Chase did their experiment and things like that. Okay, so R strain transformed to S strain and became virulent. This is the answer. Not R strain transformed to S strain and lost virulence. No, virulence. R strain was never virulent at the first place, right? S strain transformed to R strain and became virulent? No. Okay. S strain is already virulent, right? Now S strain transformed to R strain and lost virulence? No. Only this is your answer. Now going ahead with the next thing. If in a pond there are 20 lotus plants last year and through reproduction 8 new plants are added, taking the current population to 28, calculate the birth rate. So how many plants were there initially? Initial population size is what? Initial population size is 20. How many new birth is there? How many new birth is there? Births are there. So it is 8 of them. So what is the formula for finding the birth rate? If you want to find the birth rate, what do? You divide new birth divided by the initial population size you have had. That is almost going to give you what? 0.4. Yes, 0.4. So can I say, okay, taking the current population 28, calculate the birth rate, 4 offsprings per year, 0.4 offsprings per lotus per year, 2.5 offsprings per lotus per year, 25 offsprings per year. What is your answer? It is 0.4 offsprings per lotus per year. This is what your birth rate, my dear champions. Okay? Do not do any mistakes up there. If you want to read the answer once more, you can. To find the birth rate per individual, we divide the number of new individuals by the initial population. So birth rate is number of new plants divided by initial population by 8 by 20, giving you a birth rate of 0.4. So the birth rate is 0.4 offspring per lotus per year. Okay, so that's all. Now, pear-shaped biflagellate zoospores are found in. It's pear-shaped and it has 2 flagella. These zoospores are found in Chlorophyceae, okay, that is the green algae, Phaeophyceae, the brown algae, Rhodophyceae, the red algae, or both A and B. What do you think is the answer for this? Chlorophyceae, Phaeophyceae, Rhodophyceae, both A and B. So pear-shaped biflagellate zoospores is a unique feature found in Phaeophyceae, that is what, my dear children, the brown algae. It is the brown algae. Please remember that. And that is what you find it over here. Okay? Now, the inflorescence with ray florets and disc florets is seen in. Ray florets and disc florets, as in, have you seen the sunflower? They have in middle something like this. Small floret, some sort of thing. And then you have this. So basically over here, this thing that you see is the disc floret. It is what? Disc floret, which is the floral parts which is there on the disc, my dear children. If you see properly, you will be able to find it. And this petal-like thing that you see over here, they are known as ray floret. They are known as ray floret. So where do you see? Sunflower is a part of which group, my dear children? Malvaceae family or Solanaceae family or Asteraceae family or Fabaceae family? Your answer is Asteraceae. So you find ray florets and disc florets in Asteraceae. Cool? Yes. Going ahead, UTR is or are. Untranslated region of mRNA, it is present only at the 5' end. They are required for efficient translation process. It is present only at the 3' end. What is it? So it is actually the untranslated region of mRNA, my dear children. So this is the mRNA, suppose. So there will be untranslated region over here, the 5' end, okay, and also after the 3' end. So both the areas you will be finding this untranslated regions. It is present only would be wrong. Okay, they are required for efficient translation process. Yes, my dear children, they are definitely required for efficient translation process. They help in initiating and doing the other stuff. It is present only at 3' end, wrong. So can I say that this and this is the only correct answer that is there? Yes, it is there. So for initiating translation only, you need UTR and things like that. So it is very, very, very important. Okay, also for ending it up properly. Now untranslated region of mRNA, and they are required for efficient translation process, which makes 1 and 3 as the only correct option. 1 and 3 are correct. Okay. Going ahead, any species growing exponentially under unlimited resource condition can reach enormous population density in short time. Then the population growth curve will be hyperbola, J-shaped, S-shaped, and none of these. Can you please try to solve this? I will be back in a second. Okay. Remember over here it is said that there is unlimited resource. Okay, and it is growing exponentially. So obviously it is showing exponential growth, and an exponential growth curve. Which type of curve is there? You tell the answer, I will be there. Okay, I will be coming back in a second. Okay, so yes, we were in this question. Let's see. So over here, I am saying there is exponential growth, that is going up. So obviously this type of curve that you say, it's not hyperbola, it's J-shaped curve, my dear children. It looks like J, right? Because it is growing like that. And now if at all in the question it was mentioned that there is limited resources, in that case obviously the growth cannot be like this, so it will be almost like this. Okay, so it will increase and then it will be reaching a stability point. Okay, you call this point as asymptote. You call this as asymptote, and that is a S-shaped curve found in logistic growth. So logistic has an S, so S-shaped curve is also found in logistic growth. Okay, always remember that. Exponential growth, there is a J-shaped curve. Cool? So this becomes your answer. Now, classification based on evolutionary relationships between different organisms is called. When you are talking about evolutionary relationship, that is probably this organism might have evolved from this, and then because they have evolved from a common ancestor, I will be putting them down into a common group and things like that. So classification can also be done on evolutionary relationship basis, right? So what do you call this as? What do you call the system as? Artificial system, natural system, phylogenetic system, or not the above? It is phylogenetic. The term phylogenetic term comes, no phylogeny, phylogenetic. Just understand you're talking about evolutionary relationship, my dear children. Okay, so this is your answer. Next, nucellar embryos are amphimictic diploids, apomictic haploids, and pentaploids. What is your answer? So what do you mean by nucellar embryos? Listen to me carefully. So you have the ovary, inside that you have the ovule. Usually you have cells around it. This is the cells of nucellus. What do you call it? These are the cells of nucellus. Now, what is happening is that one particular cell is going to divide and form the whole embryo sac. Yeah, in embryo sac, you will be having 3 antipodal cells, 2 synergid cells, 1 egg cell, and also 2 polar nuclei. What exactly happens over here is that usually all the cells that you seeing over here except the polar nuclei, things like that, okay, they are all haploid in nature. They are all haploid in nature because they have undergone meiotic division. But suppose we are supposing over here there is no meiosis happening, all of them are diploid just like any other normal cells which are there. Chalo, let that also be a different case. What exactly is happening is that usually the egg cell and the male gamete, they fuse together and form the zygote. But over here, the cells of the nucellus, the cells which are surrounding over here, okay, generally they degenerate, but over here they are going to enter into the embryo sac. They are going to enter into the embryo sac, and this bunch of cell is going to form one embryo, this another embryo, this the third embryo, and the list goes on. So you have multiple embryos being formed, and all these embryos, my dear children, can I say they are diploid because they are arising from a diploid cell, that is nucellus cell. Nucellus cell is a diploid cell, right? They are just a normal cell, right? So it is 2n. And is it normal reproduction that is happening over here? No. Do you think there is fusion of gametes and that is how embryos are being formed? No, my dear children. So you'll call this apomixis, yeah, which is why you'll call them apomictic diploids. Okay, so nucellar embryos are not amphimictic diploids, they are apomictic diploids, not apomictic haploids. They are not haploids, they are diploids because they are arising from what? Cells of the nucellus, which is right over here. Okay, not pentaploids as well. Your only answer is apomictic diploids. Okay, example, example you want, citrus fruits which are there, they are the example of apomictic diploids, or you also call this condition as polyembryony. You have multiple embryos being formed, so you call this condition as polyembryony. Okay, fine. Next, butyric acid is produced by a bacterium. Is it Aspergillus niger? No ma'am, it's a fungus. Is it Acetobacter aceti? No ma'am, it's a bacteria, but it is not the one that produces it because it produces acetic acid. Ma'am, is it Lactobacillus? It is actually Lactobacillus. Lactobacillus produces lactic acid. No, this is not that bacteria also. It is Clostridium butylicum, the one that has close resemblance to the name of butyric acid as well. This is the bacteria that produces butyric acid. Always remember, Clostridium butylicum produces butyric acid, Acetobacter aceti produces acetic acid, Aspergillus niger produces citric acid. That is something difficult to remember because it does not have any resemblance to the name, and it's a fungus that produces it. Always remember that. Lactobacillus produces lactic acid. Okay, going ahead. Which of the following hormone in combination with auxin stimulates cell division in plants and determines the course of differentiation mainly? So, cell division is a very big term, okay, and cell division has very much to do with the name of the hormone that does it as well. It is ethylene? No. Is it IIA? No. Is it gibberellins? No. Is it cytokinins? Yes. Cyto as in cell. Kinin means division. So, cell division is mainly carried out by whom? It is being carried out by cytokinins, my dear children. So, over here, which of the following hormone in combination with auxin stimulates cell division in plants and determines the course of differentiation? Stimulates cell division in plants and determines course of differentiation would be cytokinins. Okay, ethylene is basically a ripening hormone that is used for fruit ripening and stuff like that. Okay, and gibberellin basically, which is there, it helps in germination to happen. Okay, and it prevents abscission and stuff like that. It helps in stem elongation. Okay, so yeah. Next, the cells that are found in between the xylem and the phloem of dicot root are, in between the xylem and the phloem, what is there in the dicot root? Parenchyma, collenchyma, chlorenchyma, sclerenchyma. So, I will tell you from here. Sclerenchyma is something that gives toughness, rigidity. Do we need it there? No. Chlorenchyma, which is there, it is a type of parenchyma only which has chloroplast in it, which is very much important for photosynthesis to happen, my dear children. But within the plant, do you think photosynthesis would be happening there? There is no element for basically taking up the light. You get it? There is no point of having it there. It can be there in the leaves and stuff like that. Okay, so I will say this is also not the answer. Collenchyma, again for flexibility and stuff like that, we have collenchyma. Parenchyma is a normal cell which can be used as fillers between different type of tissues and stuff like that, which is why it is parenchyma which is there between xylem and phloem of dicot root. The cells that are found in between xylem and phloem of dicot root are what? Parenchyma, my dear children. Okay, works? Chalo. Next, isolated protoplast from two different varieties of plants, each having a desirable character, can be fused to get hybrid protoplast. These hybrids are called as. What do you mean by protoplast? At first, you should be knowing that. So, suppose this is a cell, a plant cell. This is also a plant cell. Plant cell has cell membrane and also something that is known as cell wall. Agreed? It also has cell wall. Now, if I am removing the cell wall, wait, before that, I want to say a lot of things over here. So, what is this structure, my dear children? This is the nucleus. Inside it, I call this whole thing which is there, it is nucleoplasm. What is over here? It is cytoplasm. Cytoplasm plus nucleoplasm is protoplasm. Is what? Protoplasm. Ma'am, but here protoplast is asked. What is protoplast, ma'am? Basically, in a plant cell, if I remove the cell wall, whatever is left behind, including the cell membrane, it will be known as, whatever is left behind including cell membrane, it will be known as protoplast. So, basically in this type of hybridization, what I am doing is, I am removing the cell wall and making the protoplast come together. So, what do I call? What do I get? Somaclones, somatic hybrids, gametic hybrids, cytoplasmic hybrids. It is somatic hybrids that I get over here. Always remember. Pomato. Okay, potato and tomatoes fusion, pomato. So isolated protoplasts from two different varieties of plants, each having desirable character, can be fused to get hybrid protoplast. These hybrids are called as not soma clones, but they are known as somatic hybrids, not gametic hybrids, not cytoplasmic hybrids. Okay, just somatic hybrids. Cool, cool. Next, reduction of NAD does not occur in which of the following? Isocitric to alpha ketoglutarate acid, there reduction happens. NAD converts into NADH, yeah. Malic acid to oxaloacetate, here also reduction happens. NAD to NADH, yeah. Pyruvic acid to acetyl CoA, here also reduction happens, right. Succinic acid to fumaric acid, here it is not NAD which is getting reduced, but instead it is FAD that gets reduced to FADH2. Always remember in the case of fumaric acid. Okay, succinic acid to fumaric acid or succinate to fumarate, my dear children, this conversion happens, not this one. Okay, so here I have a picture for everything. Isocitrate to alpha ketoglutarate. Okay, you're not able to see, I will show you. Isocitrate to alpha ketoglutarate acid, it is NAD getting reduced over here, right. Similarly, alpha ketoglutarate acid to succinyl CoA, it is happening, and then over here the option which is not there, succinate to fumarate, my dear children, it's FAD that is getting reduced, and in the case of pyruvate to acetyl CoA, as you can see, NAD is, okay, if you're not able to see, NAD is reduced to NADH. This is pyruvate and that is acetyl CoA. Cool, cool, ma'am. So your answer for this question would be this. Okay, next. Which one of the following pair is not correctly matched? Abscisic acid, stomatal closure. Yes, abscisic acid is a stress hormone, my dear children. So if you keep your stomatas open, what will happen? The water will be lost and things like that will be happening. Do we need that? No. Is it good for stress, handling stress? No, my dear children. If you're losing water, it adds more stress to the plants. So abscisic acid is a stress hormone which will be leading to stomatal closure because we do not want more stress to happen. So it is released when plant is in stress. It is released when plant is in stress. Okay, understand that. Now gibberellic acid is responsible for leaf fall. No. Gibberellic acid, gibberellin, which you call, it's responsible for germination, it's responsible for stem elongation, but not leaf fall. It is against abscission. It does not want the leaves to fall, my dear children. Understand that. So this is wrong. Cytokinase for cell division, I already told you it's there. 2,4-D, which is a synthetic form of auxin, my dear children. It is going to act like a herbicide. Yes, it does act like a herbicide as well to selected organisms which is there. So what is the only incorrect match that we have over here? It is gibberellic acid and leaf fall, my dear children. Rest all of it, they are correct. Okay. Next question. 3-PGA, that is 3-phosphoglyceric acid or phosphoglycerate, is first stable product in carbon reduction cycle, photorespiration, light reaction, all of this. Carbon reduction cycle, or you also call this Calvin cycle if you remember, yeah. If you remember, you have RUBP, ribulose bisphosphate, yeah. What happens to ribulose 3,5 bisphosphate? It gets converted in the presence of CO2 to what? 3-PGA. That's the first stable product that you get, 3-phosphoglycerate and then acid, and then it converts into the next thing that is there. Okay, so the first stable product which is there, it is 3-PGA, and that is a first stable product of carbon reduction cycle because there the carbon dioxide is getting reduced. Okay, cool. Yes, ma'am. Next, in plants, glucose is derived from, where do you get glucose? From lipase. Lipase basically is an enzyme that breaks down lipids, my dear children, and when you break down lipids, it's kind of rare to find glucose in there. You might find a lot of other things, and later if you go on dividing things again and again and again and again and again, probably you might get something out of it, but not exactly this. Yeah, talking about sucrose, it's a disaccharide made up of two, sucrose is made up of two sugars, right? So when you break it down, obviously you are gonna get glucose. Glucose is one unit. Glucose is what? A monosaccharide. When two different monosaccharides come together, you get sucrose. Okay, you get sucrose. So basically over here, in this particular case, when you break down sucrose, you get glucose because sucrose is made up of one glucose as well. Talking about storage carbohydrates, what do you mean by storage carbohydrate? Storage carbohydrate, as in I am talking about starch, my dear children. Yeah, Starch. So starch is made up of what? Starch is a polymer of glucose. Okay, it is a polymer of glucose. There are so many glucose molecules which comes together to form starch. Just give me a second. I'm getting a call. Can't do anything about it. So what will be your answer, my dear children? Is it B or is it C? Is it B or is it C? Can I say it's both B and C? Yes, I can definitely say that it will be both B and C, and I hope you got it. Why? Because storage carbohydrate is starch, and starch is made up of number of glucose molecule coming together. Sucrose, we will talk about that. It is made up of two sugars, one is glucose and the other one is fructose. So when you are breaking down fructose, you're gonna get at least one glucose for sure. So in plants, glucose is derived from sucrose, and this, which is why your answer will be both B and C. Not lipase, because lipase is going to break down, it's an enzyme that is going to break down lipids. Okay. Now, the oxidation of one NADH2 yields what? How many ATP molecules? One NADH2 yields what? I'll say roughly 2.5 ATP, but for theoretical exam purpose and things like that, we definitely say 3 ATP molecules. Not 18 ATP, it's not 6 ATP, it's not 2 ATP also, it's 3 ATP molecules. So oxidation of one NADH2 yields 3 ATP molecules. Okay? Going ahead, biofertilizers includes what? Nitrogen fixing bacteria? Yes, the bacteria that fixes nitrogen. Basically, when you say fixing of nitrogen, isn't it increasing the quantity of nitrogen in the soil? Yes. So won't the plants would be able to utilize this later on if they need it? Yes. So they are acting as a biofertilizer, right? So nitrogen fixing bacteria such as rhizobium and stuff like that. Now nitrogen fixing cyanobacteria, this is a whole different group. For example, you have oscillatoria and things like that, nostoc, annabena. They are nitrogen fixing cyanobacteria. They are also helpful. They are also going to act like biofertilizers, because yes, you are increasing the soil quality by putting in more nitrogen in there, right? Now mycorrhiza, do they act as biofertilizers? Yes, they do. What is mycorrhiza? Mycorrhiza is basically a combination of fungi and plus what? Plant root and plant, my dear children. What exactly happens over here? This fungi belongs to the genus Glomus. It belongs to the genus Glomus, and what is exactly doing? With the help of its hyphae and structure like that, no, it goes deep into the soil and will be taking phosphorus from the soil. When you take phosphorus from the soil, you can give it to the plant. What will plant give it in return? Plant will be doing photosynthesis and giving some organic compound to the fungi for its growth. Okay, so can I say A, B, C, all of them are correct? So all of this is correct. So biofertilizers includes all of this. Okay. Now, in DNA molecule, which of the following base pair is present? Cytosine and adenine, adenine and thymine, adenine and guanine, and cytosine and thymine. What is your answer for this? Which of the base pair is present in the case of DNA? So in the case of DNA, we have two base pairs. A base pairs with T, my dear children, okay, and G base pairs with C. Okay, here you have two bonds, two hydrogen bonds. Here you have three bonds. Always remember, G has close resemblance to C, which is why they are almost lookalikes, which is why they are more closely bonded with three bonds. Okay, so adenine and guanine, they are together known as purines, and thymine and cytosine, they are known as pyrimidines. They are known as pyrimidines. Works? Does it work for you? Yes. So my dear children, if you're asked about RNA, what will your answer be? So in the case of RNA, they do not have thymine, instead they have uracil, and G still base pairs with C. Always remember that. Okay, this is for RNA. Now, in DNA molecule, which of the following bases are present? Cytosine and adenine? No. Adenine and thymine? Yes, my dear children, adenine and thymine. Not adenine and guanine, not cytosine and thymine. Always remember AG. AG, come here. Okay, so basically that counts in purines. Okay. Now, identify the incorrect statement regarding Bt cotton. What is Bt cotton? Bt cotton is basically where I am going to put a gene, Bt gene. Okay, where I am getting the Bt gene, or basically the cry gene. I am getting it from a bacteria whose name is Bacillus thuringiensis, Bt. Okay, so from Bacillus thuringiensis, I am going to take up a gene, that is cry gene, which is responsible for production of toxins. And these toxins, what do? Basically, when an organism eats this particular bacteria containing the toxin, which is inactive in the bacteria's body, but once this bacteria is inside the insect's body, my dear children, what happens is that the toxin, with the help of the alkaline pH in the insect's gut, my dear children, it will convert into active toxin, and it is going to cleave down all the things up there and gonna kill the insect. So that is what it usually does. Okay, so what we are going to do, we are going to take the gene and put it into the BT cotton, sorry, in the plant, in the cotton plant. So it becomes BT cotton. Okay, now BT toxin gene has been cloned from bacteriophage, wrong. Do you think so? No, my dear children. Where do you get the BT toxin gene? From BT, Bacillus thuringiensis, that is a bacteria, right? So that is wrong. It releases and who is responsible for putting this gene into the plant? It will be Agrobacterium tumefaciens. There is another type of bacteria. Okay, I am going to this is the one that usually infects the plant. So I am going to take the part that infects and I am going to put something into it so that it goes and tries to put this particular gene of interest into the plant that we are looking forward for. Okay, so that is like a vector. Agrobacterium tumefaciens is like a vector, the one that transports the gene. Okay, where I am getting the gene from? From Bacillus thuringiensis. So anyway, the statement is wrong. Now it releases toxin that causes swelling in the gut of insect that ingest its leaf. Yes. It is transgenic plant. Yes. Cry1AC and Cry2AB genes are introduced to make them resistant against bollworms. Yes, my dear children. So there are different type of Cry genes. Always remember, you have Cry1AC and Cry2AB. This is for specifically for cotton bollworms. And then you have Cry1AB. This is for corn borer. Okay, so how do I remember it? First letter is C, so cotton. Second letter is P, so bollworm. So since there are two of them, it is known as cotton bollworm. It's very big. Here also, first letter is C and last letter is P, so corn borers. So both of them are CB, ma'am. How do I remember it? As I told you, cotton bollworms is pretty long, pretty big, which is why you need two genes for that. Here you just have one. Okay, you can remember like that. So Cry1AB comes over here, Cry2AB comes over there. Okay, so second letter is B. You can remember like that. So what is not correct? This is the one that is not correct. Going ahead. In the given figure, if C is the DNA collected from the crime site, okay, and A and B are the samples from suspect, then who is the criminal? Let's see. This is interesting. C is the thing that we got from the crime site, and A and B also we have. Whom do you think would have committed the the crime? Even you can be detectives now, my dear children. It's that easy. Even me also. Okay. So basically, you can see that DNA is kind of same over here with A and B. Sorry, with B and C. So I'll say B is the one that is the culprit, not A1. Not A. It's just B because you have the same pattern of arrangement and stuff like that. Okay. Ouch, my hand is paining. Yes, so the given figure, if C is the DNA collected from crime site and A and B are the samples from the suspects, then who is the criminal? It is B. It is B. This one. Okay. Now, to calculate the current R value for a human population, we need to know about what? R is what? Intrinsic rate of natural increase. R is what? Intrinsic rate of natural increase, that is denoted by birth rate minus death rate. Birth rate minus death rate is equals to what? R, or intrinsic rate of natural increase, my dear children. So to calculate the current R value for human population, we need to know about birth rate as well as death rate. So can I say both A and B are the answer? Can I say that? Yes. Now the carrying capacity. I should not know about the carrying capacity. What do you mean by carrying capacity, by the way? So basically, I have a population over here. Okay. There is only limited resources, which is why it can go only up to this particular level. It can grow only up to this particular level. What is this? This is the carrying capacity of the population. Okay. So D is your answer. Fine. Next question. 30th. Dash hastens or increases or speedens up fruit ripening in tomatoes and apple. I told you there is a fruit ripening hormone that we have. It is known as ethylene. Now over here, what do we have? Auxin, abscisic acid, ethiphone and gibberellin. Ma'am, ethylene is not there. But ethiphone, which is there, it is actually a synthetic version of it. Yeah, you know ethylene is a gas. I hope you know that. Ethylene is what? A gas. So the phone is a substance, synthetic substance that can produce this gaseous hormone, that is ethylene, my dear children. So I can say this is your answer. Okay, easy, no? Next, match the column. Okay, so over here E. coli, bacteriophage DNA, haploid content of human DNA, everything is given. So the one thing that I am pretty sure about, I will be going ahead with that first. Haploid content of human DNA. Okay, so diploid content is 6.6 into 10 to the power 9. Haploid content would be half of it, that is 3.3 into 10 to the power 9. Agreed? Okay, so can I say D goes with 1? D goes with 1. Yes, let's see who has D goes with 1. So I have two options, this and this. Rest of the options I'm gonna eliminate. Now I am pretty sure about E. coli DNA also, which is given in my NCERT. It is 4.6 into 10 to the power 6 BP. 4.6 into 10 to the power 6 pp. So that is C. C goes with 2. C goes with 2. I have this option that goes with that. And hence this would be my answer. This is going to be my answer. I am going to explain everything, don't worry. So talking about what do you call delta 174 DNA, if you talk about that, it will be 5, it has total amount of bases as 5386. So this is your answer. Okay, if we talk about lambda phage DNA that we have over here, my dear children, it will be what? 48,502 base pairs. So basically your answer would be this itself. Okay, always remember E. coli has 4.6 into 10 to the power 6 base pairs. Haploid content of human DNA would be what? 3.3 into 10 to the power 9 base pairs. Going ahead, the cell wall of plant consists of lignin, suberin, protein and lipid. Suberin is part of a Casparian strip found in the endodermis, my dear children, mostly. It's not found in the cell wall. Lipid, again, yes, some lipid content is there for sure, but lipid mostly is a part of cell membrane, not the cell wall. I'm not saying it's not there in the cell wall, it is there, but not very much. It's not a prominent thing which is there. So I'll say this and this is there, but I'm pretty not sure about this, and it's not Shawnee, it's not there, and this is very less. It is very less, so I can eliminate this option. Now hemicellulose, cellulose, tubulin and lignin. Tubulin is a part of cytoskeleton. Tubulin is a part of cytoskeleton that provides the perfect shape for your cell and stuff like that. So tubulin again cannot be over here. It is not a part of cell wall. Lignin, hemicellulose, pectin and lipid. Over here I told you this is something that we are not. It's not something that is very prominent. Okay, I told you it's a part of a cell membrane, but cell wall very less. Okay, proteins, hemicellulose, pectin and cellulose. Yes, my dear children, this makes up the part of cell wall. Okay, cell wall consists of all these things that you are seeing over here: proteins, hemicellulose, pectin and cellulose. Now 33. Biological oxygen demand is a measure of what? Biological oxygen demand is a measure of what? Who is demanding for oxygen, my dear children? It is who is demanding for oxygen, who is crying out for oxygen, my dear children? It is none other than the aerobic bacteria. The aerobic bacteria who needs oxygen. Why? For what? For breaking down organic matter. For breaking down organic matter, since they are aerobic bacteria, they will be needing oxygen. So that means if there is too much organic matter over here, there is too much need of oxygen. If there is too less, there is too less need of oxygen. So biochemical oxygen demand, if it is way too much, if the demand is way too much, that means probably the water body that you're talking about might be polluted. It might have high amount of organic matter, which is why you need high amount of oxygen for breaking it down as well. Okay, so biological or biochemical oxygen demand, it's not biological, actually it's biochemical oxygen demand, is a measure of what? Industrial waste poured into water bodies, extent with which water is polluted with organic compounds. Okay, amount of carbon monoxide inseparably combined with hemoglobin, amount of oxygen needed by green plants during night. It is extent with which water is polluted with organic compounds. That is BOD is a measure of that. To what extent is the water polluted with the organic compounds are there? Okay, so this becomes your Answer. So until when do we do sewage treatment? Until the level of BOD goes down, until the level of biochemical oxygen demand goes down. Remember that. Next, choose the incorrect statement or the incorrect match. Parthenocarpic fruit, seedless fruits. Parthenocarpic fruit means what? Without fertilization, usually what happens, this is the ovule, this is the ovary. After fertilization, ovule becomes seed and the ovary becomes fruit, right? Now over here what is happening? There's something problematic over here. There is no fertilization happening. There is no fertilization happening. So do you think it can form the seed? No. But is it forming the fruit? Yes, ma'am. It's forming the fruit because there are certain hormones that plays a major role, which is why the ripening is happening and the fruits are forming. But the seed is still not formed. So can I call the fruit as seedless fruits? Yes. Ma'am, can you give an example of that? Banana. Banana is an example of a fruit like that. So choose the incorrect match. Parthenocarpic fruit is seedless fruit? Yes, it is. Okay, wall of fruit is testa. Fruit's wall is not testa. Fruit's wall is known as pericarp. Okay, talking about the seed wall, the inner wall is known as tegmen and the outer wall which is there, it is known as testa. Testa is the outer wall and tegmen which is there, it is the inner wall. So this is wrong. Wall of fruit is pericarp, my dear children. Wall of seed would have been correct, or seed coat. Now, edible part of apple and strawberry is thalamus. So, I hope you know that they are not true fruits, they are false fruits. Why do you call it false fruits? So, it's usually the ovary that converts into fruit, my dear children. But in the case of apple and strawberry, my dear children, the thalamus which is there, the part of a flower, okay, it plays a major role in forming the fruit. So yes, the ovary does play a major, has a role in forming the fruit, but along with that, the thalamus also joins, and which is why it becomes a part of a fruit. So edible part of apple and strawberry is what? Thalamus. Now fleshy part of guava fruit is what? Fleshy part of guava fruit. Is it ovary? Yes. In guava, it is a true fruit, which is why the ovary itself transforms into fruit. So this is also correct. What is not correct? This one. Wall of fruit is not testa. It is pericarp. Now, Eli Lilly, an American company, prepared 2 DNA sequences corresponding to A and B chains of human insulin and introduced them into plasmids of E. coli to produce insulin chains. Chain A and B were produced separately, extracted and combined by creating what? So, my dear children, in the case of human body, in our body, the insulin is being produced like this. So there is A chain, there is B chain and there is C chain. So now you call this structure as proinsulin. Why? Because it is not active version of insulin. In our body there are certain enzymes which will be cutting off the C peptide. Okay, which will be cutting off the C peptide, and now we are left with both A and B and that are joined. Okay, this happens regularly in our body and this is how mature insulin is being formed. But now if you want to produce insulin in laboratory condition, you cannot directly put the gene that is coding for the insulin protein into what? Into the E. coli. Why? Because in E. coli the machinery would not be there to cut and join all of these things. So what did we do? We'd put the gene for A in one E. coli and the gene for B in another E. coli. This produced A strand, this produced B strand, and now together we brought them, and in the laboratory condition we bonded them with what? Disulfide bonds, my dear children. With disulfide bonds. Always remember that. Okay, not peptide bonds, not ionic bonds, not H bonds. It is disulfide bonds that you had. So you might think that it's insulin, ma'am. Insulin is a peptide hormone, then how does it not have peptide bonds? Array, two insulin strands, we are bringing them together, you need disulfide bonds. In between them, I'm not asking what are the molecules in between them. Their amino acids and their amino acids are made up of peptide bonds. I'm not joking about that. I am telling about this particular thing, that A and B peptide which is there, it is joined together with the help of disulfide bonds outside the E. coli, that is in the laboratory condition. Works. Okay. Next, mycorrhizae helps in the absorption of, I already told you, mycorrhizae, remember the fungus and the plant which is there. The fungus which belongs to the genus Glomus, what was it doing? Its hyphae was getting into the soil and absorbing what? Everyone was taking in nitrogen, fixing it. But this guy is taking in phosphorus. So your answer for this question becomes phosphorus. C is equals to phosphorus. Mycorrhizae help in the absorption of phosphorus. Works? Yes. Next, in some seeds, remnants of nucellus are persistent. Example for such seed is, in some seed, what is there? Remnants of nucellus, my dear children, are persistent. That means, over here in the ovule. I told you the nuclear cells of the nuclear usually goes off, but in some it just stays there, my dear children, and it not gets in, right? Not always. That is in apomixis and things like that. But in some cases it provides nourishment to the ovule, and not the ovule, the embryo sac which is there, which is having the female gamete and stuff like that. Not the female gamete. The female gamete fuses with the male gamete to form the zygote, so it provides nourishment to the zygote. Okay. So what is this persistent nucellus known as? It is known as perisperm. First of all, you should know that. It is known as what? Perisperm. And this provides nutrition in two type of plants, okay? Both are B square. That means black pepper and you have beet. Black pepper and beet. So in some seeds, remnants of nucellus are persistent. Example for such seeds are black pepper and beet. So over here one is given, so this becomes your answer. Cool. Next, choose the correct option regarding agarose gel electrophoresis. DNA migrates towards the negative electrode. Wrong. DNA is what? Negatively charged. So basically over here you have cathode. Cathode is negatively charged. Over here you have anode. Anode is positively charged. DNA is negatively charged, which is why it will move away from cathode and move towards what? Anode. Now DNA can be viewed without staining. Do you think DNA can be viewed without staining, my dear children? No. DNA needs a stain, and that is Etherium bromide, and even while after putting Etherium bromide you won't be able to see it. You need UV light also, then only you would be able to see it. So I'll say this is wrong and this is also wrong. Now large molecules migrate faster than the smaller molecules. Obviously the larger ones would not be able to migrate really faster. It will be the smaller ones that will be able to migrate faster. So this is also wrong. Now Etherium bromide can be used to visualize the DNA. Yes, Etherium bromide can be used to visualize the DNA, but then you need UV light also. Always remember. So I'll say this is for the correct option, comparatively correct option that we have. Okay. Yes. Please remember, you need UV light. You need UV light. Okay. Now going ahead, the total number of species that are known and described in ranges between comes under the ranges between what? Known and described, okay? 0.5 to 1 million, 1.1 to 1.2 million, and 1.7 to 1.8 million, and 2.5 to 3 million. Your answer is this, 1.7 to 1.8 billion. Total number of species that are known and they are also described. Now, inside the pollen grain, the bigger cell with abundant food reserve and irregularly shaped nucleus is what? So, if we talk about the pollen grain, my dear children, you have two types of cell. One is the big cell that is there, which is known as the vegetative cell. Okay, and in its cytoplasm lies another small cell which is known as the generative cell. So this has irregular shaped nucleus, and this would be having a spindle shaped nucleus right over here. This generative cell is the one that will be dividing further to form what? Two male gametes. Okay, so inside the pollen grain, the bigger cell with abundant food reserve and irregularly shaped nucleus is known as vegetative cell. B. Okay. Now, the segment of DNA coding for polypeptide is known as segment of the DNA that codes for the polypeptide. What do you call this? Cistron, recon, muton, proton. Your answer is cistron. You might have heard about monocistronic, and polycistronic gene. Yeah, monocistronic means they just have one cistron that is going to code for only one gene out there. Okay, polycistronic means multiple. In a particular cistron, you will be having multiple genes out there, or you have multiple cistrons, multiple genes in one particular structural gene unit. That means there's a structural gene unit over here. There's one gene, another gene, third gene, and things like that. This is known as polycistronic. Okay. Otherwise, in structural gene, there is only one structural gene codes for one particular protein. This is known as monocistronic. So segment of DNA coding for a polypeptide is cistron. Now 42. At present, we know that more than dash restriction enzymes that have been isolated from over dash strains of bacteria. So restriction enzymes would be obviously more, and the number of bacteria would be comparatively lesser, obviously. Yes. So at present, we know that more than What is the answer? 900 strains have been taken from 900 restriction enzymes have been taken from 230 strains of bacteria. Okay, so 900 restriction enzymes from 230 strains of bacteria. This is your answer, nothing else. So B is your answer for this question. Okay. Next, the diagnosis that is based on antigen-antibody interaction is called as. The diagnosis that is based on antigen-antibody interaction, what do you call this? My dear children, always remember. So you have antibodies already present over here. We know that these antibodies are specifically made against antigens for HIV, suppose. Okay. So I know that a patient that is having HIV/AIDS would be having antibodies, these type of antibodies in their body. Now what do? Basically, I cannot extract the antibodies. Instead, I'll take their blood sample and I'll treat it. The antigens, if they come and match over here, that means they are specific. Okay, they are specific to these antibodies. That means basically the person from where you have taken this from is positive for HIV because they have the antigen specific to the antibody that I already have over here. You get it? And for viewing it, again I will be having another antibody, and this antibody, my dear children, that comes in, this antibody that I am going to add is going to be linked with an enzyme. Okay, and it will come directly attached over here. Now when I add the substrate, it will change the color because of this enzyme acts, and basically there is a color change that is happening. So diagnosis that is based on antigen-antibody interaction is serum analysis, no. ELISA, yes. Enzyme linked immunosorbent assay, that is ELISA. Is it PCR? No. Is it MRI? No. Your answer is this. Okay, the diagnosis that is based on antigen-antibody interaction is ELISA. Always remember that. Now, PCR, I know, I'll tell you when does it plays a major role. First of all, amplification of DNA, you know that it is very much important for that. Apart from that, there is one big role of PCR. If we have very trace amount of some organism in our body, some pathogen in our body, basically with the help of PCR, we can amplify it and we can be sure that we have an infection because of this guy up over here. So even for finding the trace amount of things, no, we can use PCR. Now, ecosystem is self-sufficient, self-maintaining dynamic system in which there is what? Non-cyclic unidirectional flow of energy. Yes, it is not cyclic. It is only unidirectional flow of energy from the sun to the plants, from the plants. So it is non-cyclic unidirectional flow of energy which is there. That is correct. Cyclic exchange of nutrients. Yes, nutrients and matter, they have the cyclic exchange. Okay, from one organism eats another organism, as another organism, this organism dies and again comes back to soil, and then cyclic flow is there. So this is correct. Communities and abiotic components function together. Yes, biotic and abiotic agents function together in ecosystem. So can I say all of the statements are correct? Yes, ma'am. You might be asking, ma'am, what type of question is this? This is very easy. Yes, this is easy. But children, they get confused with this non-cyclic unidirectional flow of energy. Is it true? Isn't it cyclic? And things like that. But it's not. It is non-cyclic. Okay, please remember that. Now, the last question which is out there, which is not true for monarans, my dear children. They reproduce by mitosis and under favorable conditions by producing spores. I have mostly heard that it is during unfavorable conditions that an organism switches to production of spores, my dear children. And bacteria, the monarans, my dear children, they rely on fission method for reproduction rather than mitosis. Okay, so I'll say they reproduce by fission and under unfavorable condition they produce what? Spores. So this is wrong. What is not true? This is not true. But let's go ahead. Bacteria are the sole members of kingdom Monera. Yeah, most of them have cell wall. Yes, except one. The name of that particular bacteria is what? It causes pneumonia also. Can you say? Can you tell the name of the bacteria that does not have the cell wall? In the comment section, it starts from M. It starts from M. Okay, now they are prokaryotic. Yes, my dear children, they are prokaryotic. They do not have membrane-bound cell organelles. They do not have a proper nucleus and things like that. So what is your answer for this question? A. And with that, we complete this FLT, that is full length test as well. I hope it helped you. Do let me know if you have any doubts in the comment section. You can put it down. Don't think twice that what will ma'am think of me or what my friends think of me and things like that. No. And for children who are asking me, will the question come like this in NEET or in any exam? No one can actually predict the type of questions that are going to come. But yes, it's like more of concept practice and also more of time driven practice. You should be knowing that, okay, am I able to solve questions within this particular point of time or not? That is the thing. Okay, and I'm telling you, NEET or any NEET examination, it does not have too difficult questions as that you cannot answer it at all. If you have a basic understanding of a lot of concepts that is there in your NCERT, I'm telling you, you can score really well. I'm not telling you you can rank and all, but yes, you can score a decent amount. So please make sure you're going through your NCERT and things like that really well. You're having your concepts really strong. It will definitely help you out, my dear children. And as I told you, if you need any help, you can put it down in the comment section. I'm here to help you out. If you have any doubts in the question that I've solved, ma'am, can you please explain this? I will explain in the comment section, or probably if you want me, I can probably do some other video on it if you need it, but I'll do it, okay, if you ask for it. So yes. So bacteria reproduce mainly by fission. Sometimes under unfavorable conditions they produce spores. Remember, I just told you right now. Okay. So they also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to another. It is known as conjugation. When two bacteria comes and exchange their stuff, it is known as conjugation. And that's all. Thank you for watching the video, and please do not forget to subscribe to this cute little channel out there. Probably with the next year we will be bringing out some really good things out there for NEET aspirants as well. So please stay tuned. Okay, and that's all. Wishing you all the best, and please do not be worried about anything. You are going to do amazing. Bye bye. Take care.